Hello AP Biology students, let's go over the 15th section in Unit 6. And this is one of our last sections in Unit 6 where we're talking about biotechnology, and we're going to talk about a certain type of biotechnology called bacterial transformation. One of the main reasons I have a whole section devoted to this content is because there's a very important and really popular lab within AP Biology called the Piglo lab that's going to be an example of what bacterial transformation can do. Before we get into what bacterial transformation is, we have to know about restriction enzymes. Restriction enzymes are enzymes that bind to specific locations of DNA and cut that DNA in a specific manner. If you take a look at the examples above, you can see each one of these different types of restriction enzymes is going to find a specific sequence of DNA. When that restriction enzyme finds that specific sequence of DNA, it's going to cut it in a specific manner, as you can see here. Sometimes they'll leave what's called sticky ends, where one strand is a little bit longer than the other strand, or sometimes they're going to leave blunt ends where both strands are the same length. When we're talking about bacterial transformation, we're talking about prokaryotic cells, and prokaryotic cells have this extra strand or extra piece of DNA called a plasmid. These plasmids are basically just smaller forms of the chromosome that they can exchange between different bacterial cells. So again, basically, bacterial cells have plasmids, which are small circular strands of DNA. And what we can do with biotechnology is we can actually take the restriction enzymes and cut those plasmids up in specific ways. What this allows us to do then is also use an enzyme known as ligase to bind a specific gene into a plasmid. That gene can make a host of different proteins that are very useful for our lives. So by taking this one plasmid, cutting it with a restriction enzyme, adding a target gene, and then binding it back together with ligase, we can essentially give this plasmid to a bacterial cell to make. Here's again an example of what this looks like. You can see this plasmid with the restriction enzyme. The restriction enzyme site is called EcoR1. And what happens is once we cut that site with a restriction enzyme, we can add in a gene with the same restriction enzyme cut on both ends. That again allows us to bind the target gene into the plasmid. Now again, once we add this plasmid with recombinant DNA, this bacterial cell will take up the plasmid and make the gene that we want it to make. Not every cell will take in this plasmid, but the ones that do will also receive an antibiotic resistance gene. This will allow us to grow on a bacterial plate so we know which bacterial cells have taken up the plasmid. Once we have bacterial cells with the plasmid with this gene that we want, we can take them and grow them even further, making the protein that we want. Once we have a culture of this bacterial cell with the added plasmid, again, we have transformed the bacteria, we can use that protein by extracting it and purifying it and just making it a pure protein. This has given us an ability to make protein at a much easier, higher, more efficient level than ever before. For example, bacterial cells now make insulin for our bodies, which is a much better and more ethical way than what we've done in the past, where we've had to extract it from like pigs. So we're going to keep on seeing this bacterial transformation play a really important role within biotechnology and medicine.